to everybody. Um, Thanks for attending these webinars. And for those who've now attended them for some time, this is the last one in the series on COVID-19, and we've had a nice spectrum of speakers. But today it's a great privilege actually to welcome a great friend. I think it's more family and a loyal supporter of the AAPS for many, many years. So we've saved the best for last, as you can see. So I'm referring to Dr. Dee Silverthorn, who is currently a distinguished professor in Austin, University of Texas. She's at the medical school. And of course, her record speaks for itself. So many awards and honors, including the APS's Arthur Guyton Educator of the Year Award, and then of a renowned textbook, Human Physiology. I think it's about the 8th edition. She could correct me on that. So it's an absolute pleasure to welcome Professor Dee Silverthorn. Just some etiquette before we hand over to Dr. Silverthorn, just so we switch off our microphones, video, that's for myself, during the talk. And feel free to comment in the chat line, to pose your questions at the end or during the talk, and also by raising your hand. So we look forward to an exciting talk and lots of discussion afterwards. So without further ado, over to Dr. Dee Silverthorn.
Good afternoon, everyone. I am talking to you from Austin, Texas, where it is currently early in the morning. And can you see my screen now? All right. So I was asked some time ago to talk to you about coronaviruses and physiology and teaching. And so as we go into this, I want to sort of make a disclaimer. First of all, I'm not a physician, so I try to stay away from much of the clinical work in this area. And it's also an area that I am not an expert in. So what I will be speaking today about is really my interpretation of the literature and reading and the news. And much of what I say today may be completely out of date by tomorrow because the information is coming at us so fast now. I will also, so that you don't have to try and scramble to write things down, provide Kennedy with a complete handout that lists a number of the papers that I've used, most of which are available freely over the internet, and also include the illustrations that I've used for the talk. So we're going to be talking both about the virus, SARS-CoV-2, and the disease, COVID-19. And it has become shorthand in America simply to refer to both of them as COVID. So that will be the standard that I will use today. I think we're in an amazing time right now. It's completely unprecedented. All of a sudden, this disease has come up out of nowhere. It has spread wildly throughout the globe, and we don't know a lot about it. And so we now have a chance to model for students what it's like to do science. And I think for me, one of the most important things about COVID is that it can teach our students to better tolerate ambiguity. If your students are like ours, they want the right answer. Just tell me the right answer. And there is no answer here. And the answer changes as we get new information. So I think that is one of the most important things about using COVID in our teaching. There's so much we don't know, and the models, the idea that science is not fact, it is models, and these models can change, is something that COVID is perfect for demonstrating. The other thing is that it is a a disease, when we start talking about COVID-19, that affects multiple systems of the body, and we get a chance to show students how the body systems do not function independently of each other, how much interaction there is, and how a prediction based on one system may not come true because of the interactions with other systems. And then finally, each day there's something new in the news. And so it's a chance to talk to students about biomedical research. And as we are coming up on having a vaccine, the ethics of biomedical research. So there are many, many different teaching areas to explore, most of which are way beyond my expertise and not in the realm of the courses that I teach. So today we're going to focus on physiology and pathophysiology with a tiny bit of pharmacology involved. So what I'll do for today is talk about what is some of the content we should be teaching, and then particularly how can we use active learning to teach the content rather than simply standing up and giving a lecture. And depending on time at the end, we'll look at some of the specialty areas that we can go into with students and how we can use this pandemic to teach students to think about science and how to find valid information, which has been a real problem, at least in America during the pandemic. So for the content, we'll look quickly at viruses and the process of infection in humans, with the role of ACE2 being the primary focus. And then quick overview of defense, and then some look at COVID-19. So as I go, what I will be doing is showing you how I might teach it to students and some of the questions that I might pose or some of the activities that I might give them. Whenever possible, the idea is to make them think about it and let them discover things for themselves rather than simply telling them about it. The other area that's important is when you ask students to make predictions, it doesn't matter that the prediction is what happens. It simply needs to be something that is rational and supported by the evidence. Because in many ways, the predictions then become testable hypotheses that someone could go into the laboratory and start to explore. In America, it is not very common that we teach immunology and microbiology in physiology. Those are usually separate courses. So it's important when I teach that I at least give my students an overview of viruses. And probably the biggest misconception they have is that viruses are alive. You can't kill them. They are non-living particles. They are also obligatory parasites. They must get into a host cell to reproduce. And if they don't get into the host cell, then they disappear, as we have seen with SARS and the Middle Eastern virus MERS. Basically, viruses are a core of nucleic acid surrounded by a capsid made of proteins, and then some viruses have an outer envelope. And the coronaviruses that are a family are some of the enveloped viruses. The word corona means crown, and people have said, oh, well, that's because of the spiked proteins, but they were actually named on the basis of the corona or halo that shows around the sun during a solar eclipse. Because when you look at the coronaviruses under the electron microscope, they appear to be the photographic negative of a solar corona. This is an illustration of a coronavirus. It is a single-stranded messenger RNA virus. The envelope is a phospholipid bilayer that is derived from the endoplasmic reticulum of the host cell when the virus is being assembled. The proteins, however, in the envelope are not the host proteins. The virus makes its own protein, And the one that has received so much attention for the COVID virus is the spike glycoprotein, because it is the spike protein that serves as the attachment to the host cell and that triggers the endocytosis of the virus into the host cell. So I would review this with my students because many of them have never seen it. So viruses have to get into host cells to reproduce. And so the virus comes along and it binds to a host receptor. And this is the terminology that's used. And so the first question I ask my students is, why would host cells have receptors for viruses that can harm them? And the answer is they don't. Uh, we call it a receptor, but it's actually the virus having evolved a protein structure that is mimicking whatever the natural ligand is for these surface proteins. And so some of the scientific journals are actually now putting quotes around the word receptor because it is not truly a receptor. And in the case of COVID, we know that it's an enzyme rather than what we would traditionally call a receptor. But viral binding then triggers the endocytosis of the virus. It gets digested, and with the release of the viral RNA, the virus then takes over the host internal machinery to replicate itself, reassemble itself, and then is released either by exocytosis in the case of coronaviruses. In some cases, they simply kill the cell and the virus is released. So with COVID in humans, we now have a pretty good idea of how this process works in detail. You can see here in red at the surface of the cell is the ACE2 enzyme, and that then is the receptor for the COVID virus. It requires a second transmembrane enzyme, which is a serine protease shown in yellow, to activate the ACE2 and cleave the spike protein. And at that point, the virus and still bound to the receptors is brought in by endocytosis. And then you can see it going through the replication process and multiple copies being released. So when this virus first came out, my big question was, what is ACE2? We don't normally teach ACE2, and how is it different from ACE? So this is a pretty traditional figure from a textbook showing the RAS, or renin-angiotensin system. And you can see there is ACE. It's a membrane-bound enzyme found on the cell surface. And when we first learned about ACE a number of years ago, the traditional thought was ACE was found only lining blood vessels of the lungs. So the next question I might ask my students would be, why are the lungs the ideal place to locate this enzyme? And the answer is fairly simple and anatomical, which is all of the blood that comes back to the right side of the heart goes to the lungs. So you know that in one pass through the lungs, you will get all of the blood. And so it turns out that was a good idea, but now we know that ACE is found all over the body and not just in the lungs, although its presence in the lungs has been important in the development of COVID-19. So ACE then converts angiotensin 1 in the plasma to angiotensin 2, and the primary trigger for this pathway then is low blood pressure, and all of the responses of the body to angiotensin 2 are aimed at raising blood pressure. Where does ACE2 come into this? Well, you can see it here. Here we go. And it turns out ACE2 is part of the metabolism for the renin-angiotensin pathway by taking a single amino acid off of both angiotensin 1 and angiotensin 2. ACE2 then creates the metabolites, and it's angiotensin 1 through 7 that is the important one, because this peptide actually has its own cell receptor and its own cell pathway. And it turns out the effects of angiotensin 1 through 7 on the MAS receptor is responsible for a series of responses that oppose those of angiotensin 2 on the AT1 receptor. And so now what we have is a homeostatic system where the body, under normal circumstances, is going to have this balance between the action of angiotensin 2 on the AT1 receptors and the action of angiotensin 1 to 7 on the MAS receptor. So here's another example of a schematic, and what happens in these systems, and this is a good thing for students to think about, when we take the system out of balance by using drugs. The drugs that are used to treat hypertension are the ARBs, or angiotensin receptor blockers, which happen to be specific for the AT1 receptor, and the ACE inhibitors. And then now we know that ACE2 gets involved with the COVID virus. So this provides a teaching opportunity for us. So I might show my students this pathway that we just saw and say to them, what do you think will happen to ACE2 concentrations in a person who's taking either an ACE or an ARB? And give them a chance to think about it. And the idea is that if these are building, if angiotensin 1 and 2 are building up in the pathway, then you're going to see upregulation of the ACE2 to take care of it. The next question I would ask them would be, all right, now we know about SARS-CoV-2 and how it gets into cells by binding to ACE2. So predict what's going to happen to ACE2 levels in someone who's infected with SARS-CoV-2. And the answer is the ACE2 receptor is going to be endocytosed and digested along with the viral particles. So we're going to see a loss of ACE2 from the cell surface in infected cells. And then how would this affect the balance of what we see in the body? You might think that blood pressure would go up, but probably more important is the secondary effect that we often don't talk about, which is that angiotensin 2 has pro-inflammatory effects. And so as we lose the ability to break down angiotensin 2 into Ang 1 through 7, then we're going to see enhancement of the inflammation during the viral infection. And then finally, and this became quite a matter of concern clinically during the spring, which is what's going to happen to levels of ACE2 when someone is taking either an ACE inhibitor or an angiotensin receptor blocker. And this was a big concern because we know that having hypertension is one of the risk factors for COVID-19. And so if someone is taking one of these drugs, what do you think will happen? Because we just said that they upregulate ACE2 on cells, and that would suggest then that people are going to be more susceptible to the virus if they are taking one of these antihypertensives. So now we have a testable hypothesis. And what does the evidence show? That there was the Lancet article in March of 2020 that predicted that people treated with one of these antihypertensives would have an increased risk for having severe COVID. And this was of great concern. My husband is a GP, and his patients were calling him and going, "Should I stop my drugs? Should I stop my drugs?" Well, fortunately, we've learned through the summer, and again, it's mostly observational studies, but it appears that taking these drugs is not harmful. It does not increase the risk for severe COVID-19. And there are actually some studies that suggest that taking the drugs may be predictive. And why would that be? Well, it turns out that if you are blocking the AT1 receptor with an ARB, there are still AT2 receptors, and the AT2 receptors actually are along the lines of the MAS receptor in terms of causing blood pressure lowering effects. And so the action of angiotensin 2 on the AT2 receptor may be helpful then for treating the drug. But the real conclusion is we don't know. There's only observational work, and the clinical recommendations, at least here in America, are if you're taking one of your antihypertensives, keep taking it because of high blood pressure being a risk factor for the COVID-19. So, how can we use and how can we get students to think about what we know about how the COVID virus works to either prevent or slow infection? And in order to talk about this, we need to review quickly the immune system with our students because many of them have never taken immunology. So this is the very simple approach to the immune system, which is it has two components. It has immune cells and immune chemicals, and there are three stages. The first stage of protection is the barrier function. If you can keep a pathogen out of the body, that's the ideal. But if the pathogen gets into the body, then you have two responses. You have the rapid response, which is not specific. It's already there and ready to attack a pathogen coming in. And then it will trigger the slower and more specific or adaptive immune response. The barriers, the one we obviously think about is the skin, but the barriers that are the most vulnerable are the two that we find involved in COVID, which is the airways and the GI tract. So these are systems that have to balance protection with their function of getting substances into and out of the body. So there are various types of protection that form the barrier. They have the physical, which is the secretion of mucus and other secretions. And then they have various flushing mechanisms, such as the cilia of the mucociliary pathway in the lungs and airways, and coughing is a good way to flush. And then for the digestive system, a change in GI motility, so diarrhea and vomiting. And then finally, included in the secretions are the chemicals. So antibodies can be secreted, enzymes such as lysozyme, and for the stomach, the very acidic pH is protective. So these are the barriers that these vulnerable surfaces try to throw up. In terms of the immune cells, what we see are three major types. The nonspecific response that is triggered by macrophages and other phagocytic cells that will ingest, digest, and then present pathogen antigen to trigger other cells into responding. Then we have the cytotoxic cells, which are the lymphocytes of various kinds, including the natural killer cells that are specifically designed to recognize and kill virus-infected cells. So they will secrete chemicals, and the infected cell will then undergo apoptosis and disintegrate. But what happens to function? If you've just killed a virus-infected cell, you have lost function. And so this turns out to be a case where the response to the immune system is as bad as being simply infected. And then finally, we have the B lymphocytes that produce the antibodies. And so our chemicals then are the ones that are always present, such as your interferons, which play a significant role in COVID, the complement cascade, and then interleukins and other cytokines that are starting to be produced by activated cells. And then of great interest for us now are the antibodies, because the antibodies can actually neutralize the virus and keep it out of the cell. And so the idea of neutralizing monoclonal antibodies has been a critical one in the fight against COVID. So what I might do with my students then to make them think about this rather than telling them is give them this figure again and say, think of all the ways you might develop to prevent SARS-CoV-2 from reproducing, getting into and reproducing in host cells. Go step by step, and for each of the interventions, describe how you might disrupt the process. It doesn't have to be an actual process, just think of things that you might be able to do. And they're pretty good and they're quite creative with this. And so when we come back together, we would go through some of the different steps. So the first step is to prevent spike binding to ACE2 or to the serine protease. And this can be done particularly with the monoclonal antibodies. And it's hard to see in this picture, but one of the little viral cells, viral particles outside there has a blue antibody bound to it. So finding some way to block and disable the viruses so that they cannot bind and get into the cell. A second way would be to prevent endocytosis. Yes, it's bound, but is there some way to block the endocytosis of the bound particle? And there was just, this is an example of news about it. There was just a study published on ferrets where they gave ferrets a nasal spray that contained a fusion inhibitor, and it prevented fusion between the viral capsule and the host cell membrane and stopped the process of endocytosis. This figure is a general figure for all different types of viruses. So you see things like budding and viruses being released. But the reason I put it in here is to show you many of the antiviral drugs that are now working to prevent replication inside the host cell. So you see down at number 6, we have interferons, and they are generic protein synthesis inhibitors, and we know that interferons have been helpful in treating COVID-19. And then we do have the one drug that has been approved in the United States, which is remdesivir, which is an RNA polymerase antagonist that is specific for COVID. And so finding drugs that can act at any one of these steps in the intracellular process of the reproduction is a good way to try and develop a drug for COVID. And then finally, thinking of ways to stimulate different types of immune cells. So this gives students a chance to think, to be analytical, and to see where the different drugs that are being proposed are actually acting. So as we turn to the disease, COVID-19, can we predict where COVID is going to have an effect? And as it turns out, we now know that ACE2 is expressed all over the body. We see it, the COVID-19 first in the vulnerable areas, the airways and the intestines, the gut. But once COVID is there, once it's released from cells, it can be spread through the body in the bloodstream. And so now we have many different target organs that are vulnerable to the virus. So this is the quick sort of illustration of the first part of the pathophysiology, which is the virus enters into the respiratory system. And I don't know where that COVID-10 came from. Sorry, that should be COVID-19. Then it gets into the respiratory epithelial cells using ACE, and then our antiviral effects we start to see necrosis of those cells. And as we get necrosis and the effects of losing function, then people can develop pneumonia. So the American Physiological Society has a new journal called Function that is open access, and the first issue came out, and in it was a very nice article about COVID and ENaC, the epithelial sodium channel. And this is a figure from that article showing in the first column the normal response of the airways, which is your mucociliary clearance with the mucus escalator and the beating cilia, and then fluid balance at the alveoli with fluid being secreted into the alveolar airspace. But the type 2 alveolar cells using ENaC then reabsorb the fluid to keep only a thin layer there. But SARS-CoV-2 unfortunately attacks both the goblet cells that secrete mucus as well as the ciliated cells that are used for the mucociliary escalator. And so as you lose that function, now you've lost that clearance, the virus is still staying there. And this is where that symptom of a dry cough comes in. And then as the virus gets down into the airways, it also attacks the type 2 alveolar cells. And so now we start to get fluid imbalance that leads to edema and the hypoxia that is characteristic. So this has become the standard showing the timing of COVID-19, with the early infection being the viral phase. And as we know, some people never even really show many symptoms for this. And then as the host inflammatory response starts to pick up, we now start to see the responses that are the immune responses and overexpression of the immune response. So for students, one of the things for them to think about is which pathology is caused by the virus and what is a response to what is being caused by the immune response itself. So there are so many areas to explore, and we don't have time in a seminar like this today, but the idea of we call it happy hypoxia or silent hypoxia, where people are showing very low oxygen levels without any dyspnea whatsoever. The role of ENaC, as I mentioned, in pulmonary edema. And then as we start looking at the more body-wide effects, we have the hypercoagulation with all of the microclots, and then the cytokine storm. And now there's a hypothesis that there may be a bradykinin storm, which, if that's true, would be good because we do have bradykinin inhibitor drugs already developed. Myocarditis and other heart problems that people are exhibiting, GI symptoms. This picture shows you green COVID virus particles that are in the gut of a person 3 months after they had the infection. And so the virus seems to be something like the herpes virus in its ability to just sort of hang in and hide out. The neurological symptoms, such as the loss of taste and smell, and now recently there's been something linking COVID-19 with psychological disturbances. Why are there age differences? Why do we see so little COVID-19 in small children, and it ramps up in both incidence and severity as people age? And then the anatomy and the physiology of prone breathing, which has been shown to help hypoxia. And I did want to include this because this was in one of our Del Med seminars, but it's a beautiful example showing how the anatomy plays a role and why prone position, face down, is better because of the anatomy of the lungs and the smaller surface area that is found on the ventral surface. In addition to all the physiology and pathophysiology, there's so many other areas that can be explored. If you do genetics, there are all the mutations um, and looking at the variants that have evolved, and then also trying to use genetics to develop therapeutics. You've got the immune response and inflammation, the molecular biology techniques. If you teach PCR, here's an example, a chance to talk to them about the difference between the rapid test and the standard PCR. Vaccine development of other kinds of drugs. Epidemiology has been very interesting, and the design of biomedical research and the ethics. And then one of the things that's really important for most of our students is how to find and validate information. So I think one of the things that we have seen with this pandemic is a tremendous amount of misinformation. And so finding out where your students get their information is important, and teaching them to get good information. They had a report recently on our local news that older Americans go to the standard news channels for their information, but younger people are getting their information primarily through social media, which is actually pretty scary when you think about the amount of misinformation that is put out on social media. Um, Teaching them about the difference between causality and association. So there was a, a big flap early in the pandemic about the association of the incidence with ABO blood groups. And it turns out it's not the blood group itself, it happens to be other genes that are carried on the same chromosome. Uh, Research design, great chance to teach students about research design and look at papers and say, is it observational, which so many of our COVID studies are at this point? How many people were in it? Was it humans or animals? For instance, are you going to believe a study where 9 ferrets were given a nasal spray? Or would you like to have some more information before you are willing to give that to a human? And then the ethics, and we're beginning to see this because who gets treatment, where there were conditions, situations in Italy where they were having to triage who got treatment, who will get the vaccines when they're available. And then separating biological variables in these studies from the sociological variables. So here are some quick teaching ideas for this. Whatever the news of the day is for COVID, have your students critique it in terms of the science. Give them or let them find primary research papers and evaluate, is it observational? Is it controlled, randomized? Critique the conclusions, and then propose a next step study, assuming no financial limitations. Critique a social media post, and you should be able to find some that are both good and bad, and then have them compare the social media posts to what's in the scientific literature. Was it accurate? Um, One thing, since so many of you I know teach health profession students, is to have your students create patient education materials. And they can be as creative as radio announcements or videos, but how are you going to inform the lay public about accurate science and COVID? And one of the things that we're beginning to see in this country as they start talking about vaccines is come up with the counter arguments for, we call them anti-vaxxers, people who are opposed to vaccinations for any reason. And there's a tremendous amount of misinformation going around now. For instance, a friend called because her good friend was worried. She'd heard that the vaccines were going to have nanoparticles that monitored where you were and your blood pressure, and were going to be sending information back to some mysterious person who was going to use the nanoparticles in the vaccine to control you. So I think education, both for our students and for the general public, is probably the critical thing for teaching about COVID. We can teach them the science, but they need to be able to critique the science and think, as new treatments come out, think about what's what is the logic behind it, and how accurate is it. So I think I will stop there. I'm going to guess that there are maybe some questions, but I would love to have a discussion and even hear about how you all are teaching about COVID. So I'm going to stop screen share, and there.
Is that yours, Dee?
No, that's not me. I don't know who's. That's there we go. Somebody else is at
Okay.
home.
I
So
just want to say thanks to you. I think that was excellent talk. I think that was a lovely mix between teaching us the physiology and also embedding it in a pedagogical context. So I think there's a lot to discuss, and also the fact that you've brought up the almost infodemic, they call it, of misinformation that's going around, and that you've alluded that science it's a self-correcting endeavor, that we test hypotheses through investigations, and so we advance and progress knowledge, and that we also spend more time. I think that's we can explore this to teach our students to think about science and to validate and to question, and then to communicate this to society, because it's a bit out of the ivory tower, I think, where we could just be stuck in the lab. I think I'd like your thoughts that we, as COVID alerted us to take a more proactive stance in a way for scientists, physiologists to move out of the lab into the public domain. And those are points we can discuss. Maybe you can just comment on that, and then we can see if there's any questions that come up.
I think it's absolutely critical. My teaching assignment for spring 2021 is going to be what we would call a non-majors class, and it's human health and disease. And I will be using COVID and information and teaching them as basically the lay public. They will not be science students, most of them. Most of them will be in what we call liberal arts, history, English, planning to go into law or business, and making sure that they are educated consumers of healthcare and that they know how to find accurate information. And that's a major focus of this course. And I think too often we neglect that for the hard science, but we need to be aware of that in all of our students, including our medical students, who are subject to the same pressures and looking at social media as the non-science students.
Excellent. So I'm still waiting for some questions. So I just wanted to also ask you two questions. One, you've introduced now quite a bit of immunology into the physiology curriculum. How was this received by the students?
Well, they know it's important, and I am far from an immunologist, so I mean, we barely touch the surface, but our medical students take a tremendous amount of immunology. Um, I think it's really important for them to have it. So many of our drugs are based on immunology now. Immunology and genetics have become critical. I see we have a hand raised. Soga, how are you? You're on mute.
Okay.
There we go.
I'm mute. Can you hear me now?
Yes, I can hear you.
Thank you, Dee, for a wonderful lecture. Just to follow up on what Fidel has said, in what realm can we put this issue of COVID in physiology? Do we see it as an elective or something that we think as students must know and then incorporate it as a full addendum to what we try to teach them, even though some of us may not be very good immunologists? Or when the issue of ACE2 and so on, I think we can start incorporating it so that people will also try to understand how some of these pathophysiology things fit into disease patterns, especially currently with the advent of COVID in our society. I hope I've not muddled up what I'm trying to say to you.
I
Hello?
So I think particularly the ACE2, as I was showing you, we might be talking about the normal RAS pathway. I mean, that's part of what we always teach. But then say, okay, now let's look, we don't normally talk about this, but here's ACE2. You've heard about ACE2 from COVID, and giving them the questions that are really trying to teach them how to think logically. And so the happy hypoxia is another area that I would definitely put in, which is we've talked about the triggers for ventilation. How can this occur? And trying to make them come up with the whys or hows rather than me simply telling them. So I would be selective, but I think it should be it can be used just a little bit in all of our normal teaching.
Thank you very much for that answer. So we'll bear that in mind and see how we can incorporate that into some of our teaching so that we get more modern. People, our students, get used to what is current and therefore encourage them. As you said, critique, look at literature and try and distinguish between facts from fiction and stop relying more on social media and do more of looking at proper physiology and science. I think so. Thank you very much for the answer. Thank you, Dee. Nice talking to you again.
Good to see you. I'm only sorry I was not able to come in person because the meeting was canceled. But we will trust that this pandemic will end. Yeah.
Right. Any further questions? I just have one. Dee, you mentioned more on the physiology. That we know the cytokine storm is featured a lot, but the bradykinin storm. You could perhaps just expand a bit on that.
So we know that people who are on ACE inhibitors often will see an increase in bradykinins, and this is why you may take people off of an ACE inhibitor. They develop that dry cough, and so there's some theory that the disruption in the ACE pathway is causing excess production of bradykinins, and it may actually, from what I understand, be involved possibly in the hypercoagulability. But I mean, there's so many different things. If the virus is destroying endothelial cells, right there, you have a trigger for coagulation. So teasing out what's cause and effect and what's simply association, I think is really important. I see Charity has a hand raised.
Yes, good afternoon.
Good afternoon.
Yes. I wanted to find out. There's a common question that has been, asked as to when can you talk about exercise in COVID? Because when we look at other types of pneumonia, exercise come in, but at what point can we bring in the exercises?
That is a question beyond my expertise. As I said in the beginning, I am not a physician, and I have not seen, at least in our literature, much discussion of exercise. So that's interesting, and I would have to go do some research on that.
All right, thanks a lot. Thank you.
Also, it's time for us to conclude. So I'm going to briefly thank Dee, and then I'll hand over to Prof. Sofola just to bring it to a close. And just say thanks, Dee, for a fantastic talk. I really enjoyed it. I'm sure all of us did. And we wish you well there in Texas. Sorry for dragging you out of bed this early, but it's been worth it for us. So
Oh,
I hope
well,
your circadian rhythms is not too disturbed.
thank you. I've appreciated the opportunity to talk, and just wish we could be doing it in person, but we'll.
Hopefully soon, yeah. I'm handing over to Prof. Sofola to just conclude the lecture.
Yeah, I hope you can hear me now.
Yes, we can.
Hello?
We can hear you.
You can hear me now.
Yes.
Thank you very much, Dee, once again for coming to do us a favor by accepting to give a talk, as Father said, in the early hours of morning over there. As usual, you've been able to stimulate us, because it's very difficult to find somebody who can describe a very serious topic in a very simplified way. And for those of us who are very old in the profession, it's good to be able to say that we've been able to learn something and also stimulate us, because physiology looks to be a very dynamic subject. It's not like anatomy. And it keeps changing and keeps evolving. And the idea is for us to be able to evolve as things come on. And we are very grateful that we're having you with us. So being able to stimulate us to find out that COVID is not just morbid. We can derive some physiology from it and help us to look at it. And for us in Africa, where we seem to be under suffering, let me put that to use that term, in which we don't have as much pandemic level as in the US or Europe. I think part of the things is that we can also look at the physiology and see why are we responding. There are so many theories that if this is the weather, is it because we had vaccination for anti-TB? Some of the theories are going on. But I think there are some things we can look at in the physiology, especially the respiratory system, cardiovascular system, looking at some of these linkages that you have really exposed for us. And luckily, we're going to have your lecture also uploaded for us. And then some of us can take some of these points. And if we need to get back to you to collaborate, because I'm sure as many of us are worried about the level in the US, many of you in the US and Europe are wondering why we don't have as much calamity, as much effectiveness. So maybe it's due to physiology. I think we have to look at those things. Apart from the immunologists, look at the cytokine storms, and also the inflammatory thing, the dexamethasone, antiviral drugs. I think there are some areas in which we physiologists can also contribute to finding ways in which you can ameliorate this pandemic. I really thank you for all the advice, for all the suggestions you have given, all the charts, and I think if you take some of them, it will also help us in our research and see whether it can contribute also to this pandemic, which is really ravaging the world. And we'd like to thank you again, as usual, for your expert talk. And we always look forward to hearing you talk, and we hope we'll see you at our next meeting next year in person.
Yes.
And wish you all the best in Texas, and say hello to everybody, including Ralph and all our friends in the US. Thank you very much, Dee, for your brilliant lecture. I would like to hand you over to Father to close up the thing. Thank you.
Thank you very much. And if you joined late, I will be sending Kennedy a handout that has a lot of the resources that include the illustrations that I use in my slides. So you will.
Oh, that'd be nice.
Yeah, so good. Well, thank you for having me.
Thanks,
Thank you very much,
everyone.
Judith. And thanks to thanks to Kennedy and
Thanks,
Matthew,
Prof.
and I hand you over to
Popala
Father.
and Dee.
All
And this
right.
brings our series to a close, the whole series. So Dee, thanks for ending off on a high. And it's been a fantastic series of webinars, and I hope we can do this next year again. Thanks to everyone, and bye. Enjoy your afternoon or morning if it's in Texas.
Yeah.